Hi, hello everyone. This video is for January 20th and 21st. So we're going to start off with some announcements. Then we're going to do some bell work to continue practicing our Punnett squares. Then we're going to go over some notes specifically about Mendel and his laws of inheritance. So essentially what he found through all of his research. And I'm going to talk a little bit about how to work with word problems in genetics, specifically information that tells you on how to do the Punnett square. After those notes, I'm just going to give you time to practice those word problems in the practice assignment. For the announcement, I just have one to not forget that you should have completed the following. So if you are a Green Day, this was what we were assigned on Friday, because we did not have school on Monday. So you did some of this in class and the checkpoint was homework. If you are a Red Day, this is what you should have done on the previous class period, because I was not here. So please make sure that you get those done. If you were not here or if you did not watch the video, please make sure that you watch the ICYMI video for January 15th and 19th. 15th if you're Green Day, 19th if you're Red. This is going to tell you everything we did that day, explain to you how to do each assignment and where to find them. So please check those out first. If you have any questions on them, let me know. So the first thing is the bell work. So when you go to Canvas, you're going to go to our modules. And scrolling all the way down to week 3, because that's where we're in. It's going to be Punnett Bell Work 120 to 21st. So today's date. So all of the questions that I'm going to ask are going to be about this problem that's in bold at the top here. A tall plant is crossed with a short plant. Okay. The first 2 questions are just, do you know the difference between genotypes and phenotypes? So in this problem, we are given 2 genotypes. What are they? We're also given 2 phenotypes. What are they? On a separate sheet of paper or in a Google Doc, create a Punnett square. I would suggest sheet of paper if you have one available. I put Google Doc just in case you don't have those close by. But you're going to create a Punnett square. You don't have to show me the Punnett square. You don't have to turn in the Punnett square, just do the Punnett square. Of the Punnett square, answer these next 5 questions. What percentage of their offspring would be homozygous dominant? What percentage of their offspring will be heterozygous? What percentage of their offspring will be homozygous recessive? And of course, what percentage will be tall versus what percentage will be short? All of those questions, you can get the answers by looking at the Punnett square. So for this, it'll just pop up a text entry box and you'll just answer each question in that text box. So 1, 2, 3, so on and so forth. And when you're done, you'll just submit the assignment that way. And that's the bell work. After you're done with the bell work, now you should be done with the bell work. We're going to move on to our notes. Now, I did not collect any notes, or I will not collect any notes, but I always suggest writing these down. These notes are also on Canvas if you would like to take a look at them. Just like everything else, you'll go to modules, all the way down to week 3. It's going to be right underneath your bell work. So Punnett square help, that's going to be the slides presentation that I talk about Mendel and help with Punnett square word problems. The notes that we're going to go over just now. Okay, so I'm going to start off the notes with just talking a little bit more about Mendel and his genetics. So when I first mentioned Mendel a couple weeks ago, I mentioned that he was an Austrian monk with a passion for botany or plants, and he did so much research on pea plants and looked at different characteristics and how they were inherited through generations. And he developed these laws of inheritance, these things that we know to be true when it comes to genetics. So for visual purposes, this is what Mendel did. So he would take two plants with either the same or different characteristics, and he would crossbreed them. So he would make them have baby flowers. And then he would look at how those traits are passed on. So he crossed violet and white flowers. He noticed that they were all purple or violet at first, and then he crossed those two, and then he got a variety. I'll explain in those laws what he found, but he looked at so many different characteristics: seed shape, seed color, flower color, pod shape, pod color, flower position, stem height. He looked at all of those, and his conclusion are these laws. So he came up with 3, but I'm going to share 2 with you today. The first is the law of segregation. The law of segregation tells us that only one of the 2 gene copies that are present in any organism is distributed to each egg or sperm cell that they make. And which one of those get passed on to their offspring is completely random. For example, this was a visual I gave you last week. Remember that this individual's genotype is heterozygous, so it has one big dominant L and one little recessive L. This is our genotype. So each organism has at least 2, one from mom, one from dad. But only one is present in each of their sex cells, egg or sperm. So if we say that this is the female, and she has all these eggs, some of them will have the dominant allele, and some of them will have the recessive allele. Same goes for this guy over here. If he's our guy, in his sperm, he's going to have either, because he has the same heterozygous genotype, in some of his sperm, he's going to have a dominant allele, and in others, he's going to have the recessive. Okay, so only one will be in any of those sex cells. Now, which one is passed on is completely random. So it's kind of like a coin flip where you have one of 2 chances of getting a dominant allele. So a 50% chance you can give a dominant, 50% recessive. And that's in these cases. Of course, if we're looking at different genotypes, it's going to be different. But for these, you have either or. So that's why, let's say this female gave this egg to that child, and this male gave that sperm to that child, both that have dominant alleles. That would be the genotype that our babies would get. Okay, so that's why we have those different combinations of genotypes and how we get them. So hopefully this kind of explains why we have these different genotypes and why we get what we get when we do a Punnett square. The second law is the law of dominance. So this says that an organism with alternate forms of a gene, so like a heterozygote, will express the form that is dominant. So we have talked about how we can either have a dominant allele, so like a capital letter, or a recessive allele, which looks like a lowercase letter. And they can come together in different ways because of the law of segregation to give us different combinations of genotypes. Now, most everyone has gotten the idea that a dominant, like 2 dominant alleles, will give us the dominant trait, and 2 recessive alleles will give us the recessive trait. Most everyone has this down. Where some get confused is in our heterozygous. So when we have a little recessive allele with a dominant uppercase allele, which one is it? Do they follow the dominant trait or the recessive trait? The law of dominance states that if there is a dominant allele, it'll express the dominant trait. So here are a couple examples. Up here we have some cherries. The phenotypes, they can either be red or yellow. And the genotype, so here we have a homozygous dominant is red, homozygous recessive is yellow. We can kind of see that the dominant is red and the recessive is yellow. So in the case of a heterozygous where you have both forms, the law of dominance says that we will see the dominant trait, which is why we see red cherries. Okay, here is another example of this law. So here we have a Punnett square for two flowers, both of which are heterozygous. So you have one big B, one little b. And our phenotypes are purple or violet flowers. So you might notice that every single time we have a big B, a dominant allele, we have a purple flower every time. The only time we see anything that's not purple is when we have two small recessive alleles. The only time we can see the recessive trait is if there is no dominant trait masking it. And that's the law of dominance. So that should help answer the question every time I ask you about heterozygotes or organisms that have one dominant and one recessive allele as to what phenotype they show. They'll always show the dominant phenotype. Awesome. Okay, so here are some helpful tips for Punnett squares if you find yourself struggling, or just some helpful stuff once we start doing word problems, because that's where it gets kind of a little crazy. And we'll also do some examples together. So the first two slides are mostly about terms that will be helpful to know. And I've tried to really stress that knowing the vocabulary is going to be super important and really helpful. So I just put these up on here for you all. In case you forget what a genotype or a phenotype is, you can see what they are here and also some examples. Same for the types of genotypes, the different combinations. So when I say homozygous recessive, homozygous dominant, and heterozygous, do you know what I mean? So make sure that you're comfortable with these terms because we'll use them a lot. Now, here are some steps that really help you out in being successful with your Punnett squares, especially when it comes to word problems. The first two steps especially. We've done 3 through 6, but the first two are really what are going to set you out from being successful and struggling. The first step is to always read your word problem. Read every single word of that word problem. We are usually very good at skimming. Word problems are usually not very long, so please take the time to read them carefully. The second is write out all the information that you've given or you've been given to the side. So if it tells you that we have homozygous genotypes, okay, what does that look like? Then you're going to do your usual, make a Punnett square. Then you're going to put the letters of the parents on there. That's an important part. Show your work. Then we're going to fill out the Punnett square, one box at a time. And then we're going to finish with writing out all of the data and answering the questions that are asked of you. So we're going to go ahead and do an example together. Okay, so the first step is to always read your word problem. So here we have a heterozygous round seed plant is crossed with a homozygous round seed plant. Okay, second step is to write down all the information. So I'm going to note that we have a heterozygous and it's round. And then we have a homozygous, and that it's round. So in order to do my Punnett square, I need to have actual genotypes, like letters. So I'm going to pick a random letter. We're going to go with B. Okay, I'm going to use B's for this. So a heterozygous. Hetero, again, means different. So we know that this is going to have different alleles. So we're going to have one that's dominant and one that's recessive. Okay, when we see heterozygous, that should be your automatic thought. It also tells us that heterozygous is round. Then we see that we have another plant that's homozygous round seed. Now, how the heck do we know? We just know that's homo, which means that it's going to be the same allele. So we're either going to be homozygous dominant or homozygous recessive. But how do we know which one? So going back to our heterozygous round seed plant, heterozygous means that you have one dominant and one recessive again, right? So the reason it is round is because it has that dominant trait. So kind of working backwards, you can kind of assume that the dominant trait here is a round seed because this heterozygous is round and we always see the dominant trait. So you can write that off to the side as well, that the dominant trait is round. Doesn't really tell us what the recessive trait is, but it gives us enough information to figure out what type of homozygous organism we have, right? If being round is dominant, our homozygous is going to be homozygous dominant. So then we have a big B, which is also round. So that's how we figured out the parents. So I figured out that A, the dominant is round, and B, that we have a heterozygous parent and a homozygous dominant parent. Those are things that I learned from reading the word problem and using my awesome knowledge of what I've learned so far. Third step is to make a Punnett square. So we're going to go ahead and set up our awesome Punnett square. Bam. Fourth step is to write in the parents. So again, we have a heterozygous and a homozygous dominant. Then we're going to fill in the boxes. So each box is going to grab the allele that's above it, the parent allele that's above it, and the parent allele that's to the left, and they're going to drag them into that box. So this big B will be here, big B dragged over here. This small b will stay here. Big B grabbed over from the left. Big B will be brought down. Little b will be brought over. That's a big B, I'm sorry. Big B will be brought over. Little b will be dropped down, and big B will be brought over. So then, by the time we are done with our Punnett square, these are our babies. So all the ones in yellow in the boxes are our 4 individual babies. So step 6 says to write out all of our data and answer questions accordingly. So we don't have any specific questions here, so I'm just going to write out our data. So I'm going to start with our genotypes. Okay, remember genotypes are our genes, the combinations of letters that we see. So here I see homozygous dominance. And I also see heterozygous. I don't see any homozygous recessive, so I won't write those down, but that is another option if we had different parents. So these are the 2 genotypes that I see. Now we're just going to do percentages because that's what I've been kind of focusing on. So for the genotype, homozygous dominant, we have 1, 2 of the 4 individuals that are homozygous dominant. So 2 out of 4 is going to give me a 50%. And if we look at the other genotype, which is heterozygous, we have 1 and 2 of those remaining ones that are heterozygous. So again, 2 out of 4 is going to give us a 50%. And that's how you find the genotype percentages. So for genotypes, you write down every single genotype that you see and tell me what percentage is of that one specifically. We're going to end with some phenotypes. Phenotypes. Again, phenotypes refer to the physical characteristics that you see. So in this example, we definitely know that there are some round seed plants, and that is dominant. So we know that some are going to be round. But in looking in this Punnett square, is there anything that would suggest that we don't have round babies, round seeded plant babies? No. Remember that the dominant trait is to be round, and every one of our babies has a dominant allele. Therefore, we will see them as having round seeds. So this tells us that 100% of our babies are going to have round seeds. And that's the genotype percentage versus the phenotype percentage. With those, another reminder, just write down all the different ones that you see, and then tell me how many of those there are. We're going to do this one. So step 1 is to read my word problem. So in this word problem, it says, in cats, long hair is recessive to short hair. A true breeding or homozygous short-haired male is mated to a long-haired female. What will their kittens look like? Okay. Step 2 is writing down important information. So the first thing I read here tells me that one trait is dominant or recessive to another. So on the side, I'm going to look and write down what is the dominant trait and what is the recessive trait. First thing I'm going to do here. So long hair is recessive to short hair. So what is the recessive trait?
Long hair.
Yep, long hair. So therefore, the dominant is what? Short hair. Okay. Long hair is recessive to short hair. Short hair is dominant. So you've written that down. That's definitely going to help us with figuring out our genotypes. Awesome. So that's the first part, good enough for me. The second part says that we have a true breeding, which means homozygous, short-haired male. Oops. Okay. Short hair is dominant, and they also said it's homozygous. So what is this going to look like? What is my genotype for this cat going to look like? Remember, we could do this. Oh my God. So these are examples of genotypes, right? And we can use any letter. Just T was my go-to, I guess. So of those genotypes, what is a homozygous short hair going to look like? Yep. You can use any letter. So we'll use S's. That's fine. So a true breeding or homozygous short-haired male will look like a homozygous dominant. Because it's homozygous, it tells us, and we've known that short hair is the dominant gene. Okay, this takes a little bit of like critical thinking. It's not just going to give you stuff, but I have faith that we can do it. Awesome. So now we know the male is homozygous dominant. Perfect. Now the second thing is to figure out our female, because we have to figure out what to cross it with in our Punnett square. Our female has long hair. What is that genotype going to look like? Yep. Oh, see, that's hard. It has to be the same letters that we're crossing, but I'm going to change it from S's because those are really difficult because the only thing that's different is the size. So I'm going to, same idea. K? Okay, I can do that. Oh my gosh. Okay, K. Okay, sorry about that, y'all. Yes, so our homozygous short-haired male is going to be homozygous dominant, and the long-haired female is going to be homozygous recessive, because in order to get the long hair, we can't have a dominant trait, otherwise it's going to be short. So here are our two parents. Yay! We did it. So now is the easy part, because we all know how to do Punnett squares. That's like the fun part. So I'll make my Punnett square. That's step 3. Yep. And then step 4, I'm going to put my parents. So I'm going to put my guy up top and my girl on the side. These are little K's. They look weird. Okay, so just like our Punnett squares, we're gonna grab the allele from the top parent and bring it down, and then we're gonna grab from the left and bring that over. So if I bring down all the K's or all the dominants from the top, it's gonna be my first thing. And then if I bring all the little ones over, that's what it's gonna look like. So then the question that we're answering here is, what will their kittens look like? Okay, so to me, this tells me that it's asking about phenotypes. Oh, shoot. Phenotypes. So in this, we already know that there are 2 phenotypes. So we can either have short hair or long hair. So in my babies here, do I see all of them having short hair, all of them having long hair, or some having long, some having short?
Some having long, some having short.
So I understand where you're coming from with that, because you see dominant and recessive alleles. So I had someone who said both. So this is where the law of dominance comes in, right? So you have 4 that are the exact same genotype. They all have one big K and one little k. Okay, all 4 of them. So all 4 of them are going to look the same. The question is it going to be short or is it going to be long hair? The law of dominance says that every time an organism has at least one dominant allele, it's going to show the dominant trait. Because the dominant trait is short, we're going to see short-haired cats. We're not going to see any long hair. In order to be long hair, you have to have 2 recessive alleles. So all of their kittens will have short hair. After those notes, we will do some practice problems. So you'll go to modules. And then this will be at the bottom, right under the Punnett Square help slides that we just went through. And you'll click there. And I have a copy of this on Google Docs, which I would suggest using. If you're a fan of CAMI and you really liked being able to draw or write on your paper instead of typing in it, you can use that as well. It's really up to you. But you'll click here, make a copy of this document. Click yes. I have one pulled up here. And then for each problem, you're just going to read the word problem, complete the Punnett square, and just answer the questions that it asks of you. All of these you'll need throughout the entire thing, so you should refer back to it to know which is dominant and which is recessive. But just follow the steps that I've given you, read them read out the word problem, write down everything that you know, put the parent or genotypes here, fill in the Punnett square, answer the questions. Okay. And I did get some confusion with this Punnett square box. I try to make it a little bit easier to make sure you write down the parent genotypes as well. So if we have, for example, and I'm not giving you the answer here, but if we have a homozygous dominant parent, you'll write one allele in each box. That's kind of the goal. And then in each of these boxes, you'll write down the combination of those two parents. So if I have a homozygous dominant with a homozygous recessive, you'll grab one from the top, one from the left, one from the top, one from the left, one from the top, one from the left, one from the top, one from the left. And that's how you fill it out. Again, this is not the answer. I'm just giving you examples. Most of these will be just usual practice. I believe the last two kind of give you different things to figure out, still using the same information and the same idea behind Punnett squares, but maybe you are working backwards. If you have any questions about these, just let me know. And that's it. When you're done with that, you'll just turn that back in through Canvas, and that's it.